Hey students, it's Mr. Lucido. I just wanted to create a quick recording of what I did at the end of class today, which was just to connect the different models from the group work that we looked at for cellular respiration. So again, the major objective here was to get an idea of the major purpose of each phase and then to connect each phase to the overall purpose of cellular respiration, which is to make a lot of ATP, which the cell can use. Okay, so we start with glycolysis. That was model number one. I like to think of glycolysis. The name is referring to the splitting of glucose. Lysis is breaking down. And so that's what we see in glycolysis. We start with glucose. It is broken into 2 3-carbon molecules that also have a phosphate group attached called pGal. And this molecule has a lot of energy. It does require a little bit of input of energy to get to that state, but once it gets there, the reaction will proceed in an exergonic fashion so that we can capture some of that free energy into a few different molecules. And so we see pGal, when it's rearranged into 1,3-BPG, that is going to be an exergonic process. The free energy is going to be captured in these high energy electron carriers called NAD+, or the reduced version, which is NADH. There are high energy electrons that carry the free energy released in this reaction, and it gets stored in NADH. Afterwards, this molecule of 1,3-BPG is rearranged again into pyruvate, and that also releases some energy, but in this case, it's going to result in the phosphorylation of ADP, right? So this exergonic process is coupled with an energonic process, which we know is the phosphorylation of ADP. Now, this is specifically called substrate level phosphorylation because that phosphate group is coming directly from another molecule, in this case 1,3-BPG. That phosphate was added to the ADP, as opposed to oxidative phosphorylation, which we'll talk about later in this lesson. So that's part one. So pyruvate, you get 2 pyruvate molecules from 1 glucose that are going to enter the link reaction. Now, the link reaction is the transition from the cytoplasm into the mitochondria, and it's a short reaction where pyruvate gets combined with something called coenzyme A. Now, coenzyme A, if you remember what a coenzyme is, it's something that helps an enzyme function, and that's what it's going to serve in the Krebs cycle. But this process of turning pyruvate into acetyl-CoA, which has that coenzyme attached, is also exergonic. And so you're going to be able to capture some energy into NADH, just like we did in glycolysis, which we're going to need later in cellular respiration. Okay, and so the link reaction is going to produce 2 molecules of acetyl-CoA for every glucose molecule, since we entered this with 2 pyruvates. Okay, so we've got acetyl-CoA that's going to enter the Krebs cycle. Okay, now the Krebs cycle, this is going to be the main part of cellular respiration where we're going to harvest that free energy that was originally stored in glucose. And we're going to do that through a series of reactions that are going to be coupled with endergonic reactions that are going to capture the energy released. Okay, so we have our product of the link reaction is going to get connected to oxaloacetate. And as that occurs, you're going to get a molecule known as citrate. Okay, now what I want to point out here is that each one of these sections involves multiple enzymes actually helping these reactions to occur. So for example, citrate to alpha-ketoglutarate, it's actually going to involve an enzyme that's going to help facilitate that reaction. But that reaction is going to be exergonic. like we see here, right? So citrate to alpha-ketoglutarate is exergonic, and it is coupled with an energonic process here, which we've seen in the other phases, is where we take NAD plus and we capture that free energy in the form of a high energy electron to make NADH. As we go around this cycle, alpha-ketoglutarate gets rearranged into, and that is also an exergonic process that's going to result in some ATP as well as some more NADH molecules. And as we go around one more time to this third portion, we also have an exergonic process that's going to be coupled with some more NAD plus molecules being reduced, as well as this other electron carrier molecule called FAD that's going to be reduced into FADH2. Okay, now all of those high energy electron carriers are going to be sent to the last phase, which I'll talk about in just a moment. Okay, but so far we have split glucose. We've captured a little bit of that energy. We've made some ATP through substrate level phosphorylation. And then we've also harvested a lot of free energy in the Krebs cycle that is stored in high energy electron carriers, NAD and FAD. Okay, so let's talk about the last portion. So all of those high energy electron carriers that came from the Krebs cycle, from the link reaction and glycolysis are going to end up contributing the high energy electrons that they carry to what's known as the electron transport chain. Now, as you can see down here, there are our electron carriers, and we have our high potential energy electrons being contributed to this chain of proteins embedded in the mitochondrial membrane, specifically the inner mitochondrial membrane. Now, if you notice in this figure, these proteins, in addition to passing these electrons from protein to protein, are also transport proteins. And specifically, these are active transport proteins that are pumping hydrogen ions against the concentration gradient. Because if you notice, there's a low amount here and there's a high amount here. And so we know that active transport is going to require energy. Well, where is that energy coming from? Well, the high energy electron is actually going to provide the energy in order to pump those hydrogen ions across the membrane. Now, this is important because it's going to set up the main part of cellular respiration that produces ATP, which we'll see in just a moment, by creating this big gradient, of a high amount of hydrogen ions in the intermembrane space and a low amount in the mitochondrial matrix. So as that's happening, this gradient is going to allow what's known as chemiosmosis, and this is the key process that drives oxidative phosphorylation. Because as you can see, now we have hydrogen ions are going to flow from high to low concentration areas, and we know that's called facilitated diffusion. And the protein that's going to help facilitate that diffusion, since these are charged ions, is called ATP synthase. Now, what's really cool about this protein is that not only does it facilitate the diffusion of hydrogen ions, but it also physically rotates as that happens. It physically rotates, kind of like I use the analogy of a water turbine, right? As water goes is used to power that turbine, it can be used to do work. And in this case, that energy is going to be coupled with ADP phosphorylation. So the energy that's being coming from the hydrogen ions flowing through this ATP synthase molecule and causing it to physically turn is going to be used to phosphorylate ADP into ATP. And this is where we're going to get lots and lots of ATP produced. And so we call this oxidative phosphorylation. And that's because instead of that phosphate group coming from a different molecule, like we saw in glycolysis, the phosphate group is actually just on its own, not from a different molecule, being attached to ADP by this ATP synthase molecule. Okay, so what we've done there is we've gone through the major phases of cellular respiration, and we've used these models to show how they're connected. Please reach out if you have any questions, but I hope that clarified what these models show us. Have a great rest of your evening and a great holiday break.